why, what, when, and how of the first animal skeleton. So, Duncan, you are most welcome, and please take it away.
Well, thanks very much, Jack. Thanks, everybody, for joining us. I'd just like to start by, in fact, thanking a whole host of people who have supported my research. I'm going to be presenting on a whole range of different things that I've done over a number of years. So there's various people and organizations that I need to thank. In particular, I'd like to thank Mighty Fossils, who produced many of the amazing reconstructions that you're going to see for the remainder of my talk. So with that, I'd like to start the talk proper with this piece of artwork. So it's a fantastic piece of art done by John Jew. It was part of the First Impressions art trail in the museum that accompanied the First Animals exhibition. And it features Professor Trilobite giving a lecture some 500 million years from now in the lower court of the museum. Any of you that are familiar with the museum in Oxford. And he poses the question, could evolution go backwards or in circles? Now, this piece of art was inspired itself by another piece of art by Henry de la Beche that was done in 1830 called Awful Changes, where Professor Ichthyosaurus is giving a similar lecture in the future, asking a similar question. And this in itself was a pastiche, as it were, of one of Buckland's lectures. So here is Buckland giving one of his famous lectures on geology. Now, many of the artifacts that he's presenting here are thought to be now in the collections in Oxford. Now, I'm not presenting all of this to try and put myself in the same league as Buckland, or indeed that of Professors Ichthyosaurus or Trilobite, and I can prove that because this little guy here, if I draw a circle on him, that is in fact me. So the artist came to meet me to discuss this and featured me in this piece of art. But that's not even the reason that I wanted to start here, and that is because this piece of art really exemplifies the two things that I want to talk about for the remainder of the lecture. So one is the potential cyclical nature of evolution. Do things evolve again and again? And the other is, of course, skeletons. So this wonderful scene here depicts a whole host of skeletons from this strange whale dinosaur type beast that hung from the ceiling of the museum to the rogues gallery of arthropods that are watching Professor Trilobite, of course, Professor Trilobite himself. So first of all, a little bit of context. So I'm a geologist, but I'm also an evolutionary paleobiologist. So when I'm thinking about these sorts of problems, I always have two things in mind. And the first of those is the tree of life. So this is one representation of the tree of life. So there's a handy "you are here" arrow towards the top of this diagram. We are ably represented by Charles Darwin. And I'm going to present a few different versions of these sorts of diagrams throughout the talk. So I wanted to just, for those of you that aren't familiar, give you a little signpost. So this is essentially a diagram that represents who's related to who. And it's done by joining, connecting up different organisms with lines of common descent. So Darwin up there in the top, his most close relative in this diagram is the chimp. And they are joined together by these lines that meet at a node to the exclusion of their next closest relative, in this case, which is the golden line tamarind. And then we can combine these ever more inclusive groups or clades of organisms to produce a hierarchical nested tree of life, eventually including all living things, living and indeed extinct. Now, the other thing that I always try and keep in mind is time. So the Earth is very old, and we know this by examining the rocks, and they obey a general rule of thumb, which is also the same general rule of thumb for those of us who have offices that we haven't visited for several months, the large stack of paperwork that we never got to. The general rule of thumb is that the things at the bottom are older than the things at the top. And that's exactly the same for rocks. And geologists like to divide up those rocks into packages of time that we give names to. And by doing that, we can try and better understand what's going on in deep time. Now, this diagram here is a little bit skewed because it doesn't really represent the true antiquity of the Earth. And I'll tell you what I mean with a little demonstration. So this purple line towards the bottom of the diagram, this is the crucial interval that the entire rest of my talk is going to focus on. And it looks like it was a very long time ago. And in fact, it was a long time ago. So if we go back from today to T-Rex around 66 million years, if we go back from T-Rex another 66 million years, we don't even get to Stegosaurus. Stegosaurus is further back in time than T-Rex is to today. But even Stegosaurus and other dinosaurs of the early Jurassic, we need to go back another 3 times that amount of time before we get to this boundary that I'm interested in, the so-called Ediacaran Cambrian boundary, at around 540 million years ago. But if we rescale this diagram to give the true antiquity of the Earth, you'll see this purple line shot up almost to the top of the diagram. So the Earth's about 4.6 billion years old, and for the vast majority of time, there were no animals, recognizable animals with skeletons, and very little in the way of a fossil record. And that really all happened in this last slice of time known as the Phanerozoic. And it's this remarkable change from this relatively long period of stasis to oceans and ultimately the land and the air teeming with life that we recognize today that I want to try and explore a little bit more. So zooming in on that purple line, we get to the boundary between 2 periods of geological time, known as the Ediacaran, which is from around 635 up to this boundary at about 540, 541, followed by the Cambrian. Now, these periods of time are named after rocks that are of those ages. So the Ediacaran is named after the rocks of the Ediacara Hills in South Australia, and the Cambrian is named after Cambria, which is the Latin name for Wales, because there are Cambrian age rocks in North Wales, for example. Now, the Ediacaran has recently, in the last few decades in particular, become famous for this wonderful array of entirely soft-bodied organisms. Particularly famous is the site in Newfoundland in Canada that this reconstruction here is based on. So for any Canadians here, that are listening today, happy Canada Day. And if you want to know a little bit more about this period of time, you can check out the lecture from 2 weeks ago from Dr. Frankie Dunn, who told us an awful lot about what was going on during that period of time too. And the Cambrian is perhaps most famous for these weird wonders of places like the Xinjiang biota in Canada, or another, the Xinjiang biota in China, or another famous fossil site, the Burgess Shale, also in Canada. And for, as I say, weird wonders, but things that we can recognizably place into modern groups of organisms. Now, between these two quite different worlds, the Ediacaran dominated by soft-bodied things, and the Cambrian, where we see the advent of skeletons. We have this transitional period in the very latest part of the Ediacaran, typified here by an assemblage from Namibia in Southwest Africa for around just before the Ediacaran Cambrian boundary, where we still have strange soft-bodied creatures that are very hard to place on the tree of life, like this Svartpuntia, nearest to me in this diagram. But then over on the other side, we have these weird baby's rattles and tubes that I'll talk about a little bit more shortly, that are the first animals that bared skeletons. Now, this fundamentally changed 2 important things. One was the ecology of these environments, and the other was the ability of these organisms to enter the fossil record. We see a huge increase in the preservation potential of animals when they begin to build skeletons. Now, this problem of a comparison between the Precambrian, or the time before the Cambrian, where we see no animals bearing skeletons, and the post-Cambrian record has been puzzling scientists for a very long time. And so Darwin wrote in The Origin in 1859 that during these vast yet quite unknown periods of what we would now call Precambrian time, the world must have swarmed with living creatures. And we now know from finds like these amazing soft-bodied biotas that was true. And to the question as to why we don't find records, part of that is to do with the advent of biomineralization. That's animals building skeletons. So for the rest of this talk, I'd like to follow in the footsteps of another great scientist, and that is the character created by Andrea Beattie, Ada Twist scientist. This young scientist puzzled her parents by posing a series of questions. She started with the why, then the what, how and when, and by bedtime she came back to why once again. So between now and your bedtimes, whenever that might be, I hope to go through these questions, but around the origin of animal skeletons. So we'll start with the why, and why are skeletons important? So they're obviously important to these chickens in the boneless chicken ranch from this Larson cartoon, but why are they important more broadly? So you might be thinking of skeletons like these. So big skeleton and little skeleton here, and their dog Rex. You can probably tell I've been spending a lot of time at home with the kids over the last few weeks and months. These have a bony skeleton, just like you and I do. But skeletons have a much broader range of forms and functions across the animal kingdom. So here is just a snapshot of a few different skeletons from living and extinct organisms that are made of a whole range of different minerals, and also on the inside or the outside of the body. Now, if we have a look at a tree of life, so this is just a reduced version of the diagram that I showed you earlier, but in this case, it's been drawn against a time scale. And I've added in here examples of animals that bear skeletons, with some rough idea based on the number of these shell emojis as to how diverse and numerous those skeletons are. And the point that I'm trying to make here is that animals with skeletons are all the way across the animal tree of life. Lots of different animals use their skeletons in lots of different ways. So some of those ways might be to do with getting their food, acquiring their food, or preventing themselves from being eaten, or in the case of these corals on the far side of this slide, actually building entire ecosystems. So the Great Barrier Reef, for example, all coral reefs are built by animals. Corals grow these skeletons, or it may be to aid in burrowing into sand, like this bivalve mussel is doing, bivalve mollusk is doing, burrowing into the sand in the top part of this diagram. So skeletons are very diverse, and they are very important to lots of different organisms, and also very important for building the ecosystems that we see today. So what exactly happened, and when did it happen? So this is a graph of the numbers of different groups, of major groups of animals. So there's the biggest way that we divide up animals into groups are called phyla. So that's things like the mollusks or the chordates, the things that the group that we belong to. And then we can further subdivide phyla into classes, for example, mammals. And again, we've got this purple line, which represents this boundary between the Ediacaran on the far side of the slide and the Cambrian, and then the subsequent period, the Ordovician, closest to me on the slide. And you can see this huge increase in the number of different phyla and classes of animals through this interval, almost to modern levels. So the shaded out parts of this graph are what we see in the modern. And really the missing phyla and classes are, for the most part, to do with their inability to be preserved. So what else was going on during this period of time, and indeed a little bit before? So if we go further back in time to a period called the Cryogenian, so as the name suggests, this period of Earth history was dominated by ice. So there were a potentially global extent of ice for large parts of this period of time. And there was another similar event on a smaller scale, perhaps partway through the Ediacaran, here represented by this blue line. And then shortly after this glacial event, we get the first large-bodied fossils in the fossil record. So this is an example here of this reconstruction of Charnia, which again featured in Frankie Dunn's talk from a couple of weeks ago. So we get these strange soft-bodied creatures that are increasingly, the evidence is pointing towards them being animals. And then later on in the Ediacaran, towards the end, we still get these strange soft-bodied things of different forms, some very different in construction, like this Svartpuntia. But again, the two creatures that I introduced you to briefly at the start, Narmacalathus and Cloudina, they are responsible in part for this little jump in the graph where we're seeing more diversity of organisms because we get the first things that are building skeletons. So here is a Narmacalathus. So they would have been just a few centimeters at the most in length. It's strange baby's rattle shape. We're not really sure what the soft parts of the animal would have looked like, but we have the evidence of their skeletons preserved. So on the other side of this slide, you can see a cross section through a rock, and this grey, pale grey material around the edge is the body wall of that Narmacalathus. So we know it was building a skeleton. Similarly, we have this Cloudina that consisted of a series of funnel-shaped cups composed of calcium carbonate with possibly a coral-like animal living at the top of the tube. And these are interesting not just because they're one of the first things that biomineralize, but they also perhaps have the first evidence for direct evidence of predation in the fossil record. So you can see the poor Cloudina closest to me on the slide has got a hole in it. So this is thought to be the result of predation. And these organisms are believed to have started to build their ecosystems as well. So started to build the first proto-reefs along with microbial organisms called thrombolites that were building these mounds. So we have here in this example a Cloudina dominated reef fringed by Narmacalathus from this brilliant reconstruction from John Civic. So returning then to this diagram, as we step over the boundary, we see this huge explosion in the numbers of classes and phyla of animals. And most of the first record for this explosion is in the fossils of skeletons themselves. So here is a snapshot of some of the diversity of these creatures. So they've been given the Ronseal name of the small shelly fossils on account of them being small shelly and fossils. And they come in a whole range of different shapes and sizes, spines, plates, tooth-like elements, tubes. And they sit all across the tree of life. So very early on in the Cambrian, we get examples of all sorts of different creatures building their skeletons. And again, they start to build much more complex ecosystems with greater degrees of interaction above and below the sediment, dominated by things like these early sponges that started to build more and bigger structures. And eventually we get ecosystems like the one represented here in this reconstruction of the Chenjiang biota, where although everything is still a little bit weird, we can place virtually all of these animals firmly into modern groups, and we've got a whole host of different things that have skeletons, brachiopods and sponges, and trilobites, like the one that's met its end towards the beginning of this slide. So how can we use the fossil record to try and better understand this interval? So occasionally we get lucky and find completely articulated skeletons like this beautiful slug-like Halkieria from the Sirius Passet Lagerstätten in North Greenland. So in 2 weeks' time, Professor Paul Smith, the director of the museum, will give a lecture that I'm sure will feature many more examples of amazing fossils from this particular locality. But for the most part, these early skeletons are found as disarticulated bits and pieces in the fossil record. So if we can't get lucky, then we can get tooled up instead. And for a large part of my research career, I've been using a technique known as SRXTM, or synchrotron radiation X-ray tomographic microscopy, but you can think of it as getting inside these microfossils. And a general rule of thumb is the smaller the thing you want to look at, the bigger the machine is that you need to look at it. So I've been lucky enough to visit on many times this facility near Zurich called the Swiss Light Source. Now this is the image of the front door of the Swiss Light Source on the website of the organization that run it. But because I was there as a PhD student, I didn't really see it very much like this. Most of the time I saw it like this, because we run the machine 24 hours a day, so I would often have to get up in the middle of the night to attend to my fossils. But it's all for the love of the science. And once you're inside this big donut-shaped building, you go inside a little hutch, and this is the one that we spend all of our time in. It's called Tomcat, and Tomcat is, I think, wins the award for the best backronym of all time, because it stands for Tomographic Microscopy and Coherent Radiology Experiments. You can see there's been a little bit of special pleading to get that to fit. So I've got a short video here to show you a little bit more about how this technique works. So this is a model of the Swiss light source, and these flashes of light represent electrons that are being sped around a ring, and as they speed up, they release X-rays, and those X-rays are then funneled down a metal tube essentially towards the specimen. The specimen is exposed to those X-rays, and the material of the specimen absorbs them to a greater or lesser extent, and we get an complete three-dimensional internal view of whatever it is that you've put into the beam. So this is what it looks like on the inside. So you can see on the left-hand side of this image is the metal tube, but the X-rays come down. And here's me pretending to load in a sample for a photograph. So we place it on this sample stage. So here's what it looks like. So on the part of this photo closest to me is where the X-rays are collected. It then hits a mirror and get picked up by a camera. And you rotate the sample through 180 degrees as you expose it to these X-rays. So once you've loaded in your sample, you retreat through the extremely thick metal door, unless you particularly enjoy being irradiated. And then you can control the machine from these workstations on the inside. And you end up, as I say, with these beautiful three-dimensional internal models of these fossils. And it's from this data that we've been able to much better understand how these wonderful creatures grew. So on to the main question of the talk then. So we've come all the way back around, just like Ada Twist, to the why again. So why did skeletons evolve? Now, I'm too much of a chicken to answer such a big question. So I'm going to modify it a little bit and say, well, why did skeletons evolve then? So there's been many people talk about the so-called Goldilocks conditions for lots of different natural phenomena. And I think it's probably very similar to what's going on down in the base of the Cambrian. So here is a rather busy diagram that's trying to simplify all of the different features that or factors, I should say, that contributed to the origin of this burst of the animal diversity in the Cambrian. And I want to focus in on two of these factors. So they are here in the diagram. So we've got an increase in genetic complexity and animals beginning to biomineralize, to use minerals from the seawater to build their skeletons. So for the remainder of this talk, I want to introduce this idea of the so-called Biomineralization Toolkit. So this is a hypothesis that suggests that all these different animal groups that build skeletons in this period of time were already well equipped to do so by the start of the very end of the Ediacaran and into the Cambrian. And this hypothesis makes three sort of predictions of what we would expect to see. So the first is that there would be multiple independent origins of biomineralization. So that's different animal groups building skeletons in their own way but all at the same time. So let's have a look at the fossil record and see if this prediction is borne out. Here again is that time scale zoomed in on that purple line between the Ediacaran and the Cambrian. And I can add to this the stratigraphic ranges. So that is the time from which we see the first time we see a particular fossil through to the last time we see that fossil. And these are color coded for different kinds of mineral that these different animals are building their skeletons from. And I've left off the names of all of these different obscure early skeletal groups for clarity. And then we can add on to that the tree of life and see how these different animals that are building skeletons are related. And I've highlighted a few of the more familiar groups, things like the early ancestors of snails and clams and worms and crabs, even ourselves and starfish, and down at the bottom the corals and sponges. So the thing I'd like you to take away from this diagram is that very early on, in a short window of time between the very latest Ediacaran and the first few slices of the Cambrian, we see almost all of the different animal groups that have skeletons evolve them, but they're doing so in a whole range of different ways, from lots of different minerals at different times. So there are successive waves of different groups, and in between these mineralizing groups are entirely soft-bodied relatives. So the fossil record indicates to us that we have multiple independent origins of animal skeletons in this time. So the second observation that this so-called toolkit hypothesis suggests is that there should be an inherited organic scaffold, a precursor soft skeleton, and also the genes required to build those skeletons. So we can't get the genes out of extinct organisms, very sadly, but we can look at their descendants. And there's been a number of studies over the last 10 years or so that have been doing exactly this. So just one example of one of the early examples of doing this from 2010, Dan Jackson and colleagues compared the genes required to build a nacre or mother of pearl in these two mollusks. So that's the pearl oyster and an abalone, and they compared the genes with those in this purple sea urchin, or rather in the larvae of the purple sea urchin, to produce their skeletons. Now, sea urchins and mollusks share a last common ancestor some way back in the Ediacaran somewhere. We can argue about exactly when that was. And that common ancestor was almost certainly entirely soft-bodied. But the pearl oyster and the abalone have an almost as ancient last common ancestor. These two groups of bivalve and gastropod mollusks have been around since at least the early part of the Cambrian too. So what these Jackson and his colleagues found was that these two different mollusks, even though their shells appear very similar in construction, they use different genes. They've independently evolved the ability to deposit this nacre or mother of pearl. But perhaps the most extraordinary thing is they found that some of the genes that they do share, many of them were also shared with the purple sea urchin, that were being employed to build their skeletons. So they suggested that these ancestral genes must have been independently exapted. So that's the term we use for taken to use a different function into a role in building skeletons in these two different groups. And this is in essence the biomineralization toolkit hypothesis. So there's good evidence from modern organisms that exactly this kind of process was taking place. So finally then, we should see, if this hypothesis holds true, we should see in different groups, after they've independently acquired skeletons, they should gain more and more genetic control over building those skeletons and get more and more complex as a result. So if we have a look at one particular group, so the so-called shelled or conchiferan mollusks. So these are bivalves, that's clams and mussels and oysters; gastropods, that's garden snails and slugs, but also a whole a host of marine slugs and snails and their relatives, scaphopods or razor shells, cephalopods, which includes octopus and squid and cuttlefish, but also organisms with shells like the living nautilus, but extinct forms like ammonites and belemnites. And the less well-known monoplacophorans. So these are little mollusks with a single valved shell. So they have their common ancestor back down in the latest part of the Ediacaran, or the very earliest Cambrian. And it's almost, it's very likely that this organism had didn't possess a biomineralized skeleton, or perhaps possessed a very simple biomineralized skeleton. But early in the Cambrian, we see examples of all of these different groups building skeletons, and they're doing it in a very similar way. So they have the inner layer is built in a very simple fashion using the so-called lamellofibrilla, so that's sheets of fibers of minerals. And then later on, independently, these different mollusk groups develop more complex microstructures in their shell, so a so-called prismatic outer layer, and then eventually mother of pearl or nacre. And we see the same pattern in slightly more distantly related mollusks. So there's this group called the aculiferans, which today includes the chitons or polyplacophorans and the so-called aplacophorans worms. So these are mollusks without any shell at all. But in the geological past, there were many more examples of aculiferans with shells, and we see similar microstructures in these creatures as well. But what about the group that we belong to, the vertebrates? So here is a wonderful example of a vertebrate skeleton. So this is a tuna from the museum in Oxford. And like the tuna, you and I have an internal skeleton built of calcium phosphate. Now, the group of vertebrates that I'd like to focus on for the last 10 or 15 minutes of the talk are a group that's particularly close to my heart. They are the conodonts. So the fossil record of conodonts is almost exclusively made of these tiny calcium phosphate teeth-like elements. Here you can see a range of them mounted on a pin. That's how small they are. And conodonts fit in the tree of life somewhere around the node that represents all of the jawed vertebrates. So that's bony fishes, sharks, and us. So we're essentially jumped up fish, and the jawless fishes. That's hagfish and lampreys. And it's not entirely clear yet which of these two groups, the jawed or the jawless fishes, and the conodonts are most closely related to. So conodonts arise in the latter part of the Cambrian, and they are one of the most abundant and diverse vertebrate groups throughout, all the way through to their sad demise at the end of the Triassic. Now, the very earliest conodonts are had elements, these tooth-like mouth parts that sat inside this small eel-like fish. Were essentially cones of one shape or another. So I'd like to draw your attention to the scale bar just near my shoulder. So that scale bar there is 100 microns. That's 100 thousandths of a millimeter. So these are incredibly tiny little things. And there's basically two broad groups of conodonts that I want to introduce you to. So they are the so-called paraconodonts, or alongside the conodonts, and the euconodonts or true conodonts. And they're separated into these two groups based on the tissues that are inside these tooth-like elements. So the paraconodonts just have a single kind of tissue made of layers that grew incrementally by adding more and more layers to the base of these elements. And then euconodonts, or the true conodonts, that dominate basically all of the post-Cambrian fossil record of conodonts all the way up to the end of the Triassic. They have this this unique tissue called the crown, which evolved in conodonts sometime in the late part of the Cambrian. So for the remainder of the talk, paraconodonts are in blue, and euconodonts are in blue and red to represent the addition of this crown. And with some colleagues a few years ago, I demonstrated that euconodonts derive directly from paraconodonts by the addition of this crown layer. So if you strip off the crown and then break up these elements into their individual parts, all of this, of course, done virtually by using the synchrotron tomography method that I mentioned a few minutes ago. The inner part, this blue basal body of a euconodont, is indistinguishable in terms of the method of growth from a paraconodont. So we know that euconodonts evolved via the addition of this outer layer, the euconodont crown. So let's take a closer look at paraconodonts. So here are two different paraconodonts, Furnishina and Problematiconites. And what I want to point out to you is that there's quite a big diversity of different kinds of ways in which these structures are put together. So in the Problematiconites, that you can see these slices through of this synchrotron tomography data. So we have a more complex arrangement where the layers start to grow around the outside as well as just on the inside of the elements. And also in some paraconodonts, we see this so-called spheritic microtexture of these little ball shapes of phosphate that we also see in euconodonts. So here is another comparison between a paraconodont in blue and a euconodont in blue and red that are indistinguishable if you strip off the euconodont crown. And here is another example from a euconodont where we see, so this is an actual physical slice through a larger sample, which I cut through with a saw and then polished down after I set the sample in resin. And this is examining with a scanning electron microscope. And you can see these layers inside the darker grey part in the middle, which is the basal body, and the brighter part around it is this crown. And we see the same differentiation into different tissues that we see in the paraconodonts, just with the addition of the crown. So within this lineage of the earliest conodonts, we see a progressive building up of more and more complexity. So from the earliest examples with just very simple layers growing around the base, then we get more complex examples on the base and the sides, then we get spheritic mineralization, and ultimately the building of the euconodont crown itself. And this is entirely independent from the rest of the vertebrate skeleton, so things like teeth and bones of more familiar vertebrates. So even within the vertebrates themselves, we see this independent acquisition of skeletons. And if we were to examine the record of other vertebrates, we see a similar pattern of this bigger diversity of different kinds of skeletal types in the earliest examples, and then a progressive building up and constraining of the complexity of these groups. And so you may be asking, well, where did these skeletons derive from? Well, there's potentially some evidence in modern organisms to try and fill in the gap where the fossil record can't necessarily help us. So this fascinating little creature, only a few tens of millimeters in length, is called Amphioxus, and it's our nearest invertebrate relative. So if you were to, once you've joined together all the vertebrates in that tree, and then you bring along some of the invertebrates and see where those nodes meet, one of our nearest invertebrate relatives is the Amphioxus, and they are on first appearance are very fish-like. And these scientists discovered collagen in the body tissues of Amphioxus, and this is potentially this organic precursor skeleton that I said earlier would be predicted by this biomineralization toolkit hypothesis. So in vertebrates, we have examples of increasing complexity. We have examples of the potential precursor, and also examples of the molecules required to build those skeletons. Now, I could go through many more examples of this kind of thing. So I could talk about the very earliest tubular fossils that may be related to corals, have a similar fibrous microtexture. Sorry, I'll just... or I could talk more about the early mollusks that have this building up of more and more complex microstructures, or indeed in some of the earliest sponges that show a big diversity of their different kinds of skeletons very early on, before reduction into the modern groups of sponges. And in brachiopods that we see today, their earliest ancestors also display this wider range of variety of microtextures that start off with these very simple lamellar tissues. So, and there's many more examples beside that. So hopefully I've convinced you that both the fossil record and what we understand about how modern animals build their skeletons all agrees with this biomineralization toolkit hypothesis. So I agree with this idea that there must have been just the right conditions, so that the Goldilocks conditions in this period of time to allow these different independent groups of animals to utilize the tools that they'd inherited from their soft-bodied ancestors to go on to build skeletons. And I'll finish by returning to this question posed by Professor Trilobite. So, could evolution ever go backwards or in circles? And I think in a very fundamental way, it's important to say no, that this event, the Cambrian explosion, was a one-off, and it fundamentally changed the Earth and the way that organisms interact with their environments. But there is definitely examples of in different animal groups, no need to reinvent the wheel, as it were. So we can use the tools, so whether that's a genetic toolkit or an organic skeleton inherited from the soft-bodied ancestors, to do the same thing in slightly different ways. So in some ways, Professor Trilobite might be right. So I'll finish there. Before I go on to take any questions, I'd just like to pose this question of you. So it'd be really great for us if you could let us know in the chat whether this lecture has changed your opinion on the evolutionary origin of animals or added to your understanding of the topic, and if it has, how. So with that, I'll say thank you very much.
Thank you so much, Duncan, for not only a wonderful exposition of the ideas around the appearance of animals and the rise of skeletons, but also a little peek behind the scenes at how this is all done at places like the Swiss light source. Do keep your questions coming in on the chat. We've got some, I think tonight's group might be the best set of questions yet. Excellent. So, but do keep those coming in on the chat, and also do answer Duncan's question that he's put up there. If you've got an answer to that, do please put that up in the chat. That helps us to understand what impact these talks have, and hopefully provide even better ones in the future. And I'd also say I'll be popping up another link a little bit later for the next talk, which is going to be by Professor Paul Smith, who is the director of the museum. He's going to be talking about the Cambrian explosion, the rise of animals, and the way that that can be looked at through fossil sites in the very north of Greenland. So expect some stunning fieldwork photographs in that lecture in 2 weeks' time. But before I do that, I'd just say, if you have enjoyed tonight, then please do consider donating to the museum. The museum is a charity, and it relies on charitable donations for us to be able to provide not only the public spaces, the collections, the public engagement, and the research. So there's a button has just appeared for that, and in a bit I'll put up the link again for Paul Smith's talk. And now over to your questions. We've got some brilliant ones tonight. We're going to start off with a question by Mark Derbyshire, who asks, for what fraction of the age of the universe has there been life on Earth?
Oh, wow. Well, I think the universe is about 15 billion years old, but if there's any astronomers in the audience, they maybe correct me. But in terms of when life originated on Earth, so it's thought based on looking at the time it's taken since the genes in modern organisms diverge, so we can use it as a kind of clock, count the number of changes and go back in time. These studies suggest that basically once conditions on Earth permitted life, so there was a relatively stable surface with liquid water, then a life originated. So it seems that it was almost sort of inevitability of the conditions. So the fossil record suggests there's life maybe to a couple of billion years, but these other methods suggest that life probably goes back much further than that. So I think in terms of the proportion of the universe, well, that's a harder question to answer. But in terms of the Earth, it was most of the time that the Earth has been around, it's been populated by life, but not animal life. It took a long time before these more complex, larger-bodied creatures arose later in the Ediacaran.
And now we have I think my favourite question of these lecture series yet, and you've got to be careful how you answer this one, Duncan, because of course you have many of your colleagues in the audience this evening. I can see paleontologists in from all around the world. This is from Yakub, who is 12, and Yakub wants to know, what is your favourite geological period and why?
Wow, that's a difficult question, yeah, and will potentially be controversial. But those of you that know me will know that I hail from the Black Country, which is of course famous for the Silurian limestone. So I think my heart probably left somewhere on Wren's Nest in the Silurian. But I've done most of my research on organisms in the Cambrian. So it would have to be somewhere, a fight between the Cambrian and the Silurian, but I think I'd probably end up back home in the Silurian.
An excellent answer. We go across to a question from Elizabeth Parker, who asks, if most organisms were soft-bodied in the Precambrian, in the Ediacaran, what evidence other than the fossil record was used to prove their existence?
That's a brilliant question. Yeah, so for a long time, there was essentially no fossil record of large-scale, large-bodied things in the before the Cambrian, until the discovery of these soft-bodied creatures, or probably animals. Now, the occasions where you get this kind of soft tissue preservation are extremely rare. And very little is known of them. What is more common, although still relatively rare, is that organisms leave behind evidence of their movement or their feeding behaviors in so-called trace fossils, so burrows and trackways. And the very first evidence that there's complex moving animals, definite animals, it comes from the trace fossil record. So, and in fact, the boundary between the Cambrian and the Ediacaran is defined on the appearance of a certain kind of trace fossil. And trace fossils start to get more complex towards the end of the Ediacaran in a similar way to this increase in diversity of different kinds of organisms doing different kinds of things. So the other record that we can use is more indirect, which I've alluded to, I think in one of the other answers to one of the other questions, is to look at the genetic makeup of modern organisms, and we can use that to predict how they're related and also when they diverged. And there's very good evidence that suggests that animals go back into the Ediacaran or even before, based on the time that they thought to diverge. So we can bring together these all these different kinds of evidence. We don't have to rely on these exceptionally rare soft-bodied finds, but of course when they are found, they bring immense amounts of information.
Wonderful, wonderful. And we've got a question now about your the methodology of using the synchrotron, asked by Carolina, who wants to know what is the maximum size the equipment can analyze.
Oh wow, that's a great question. Yeah, so thanks, Carolina. So it really depends on which facility that you're using. So the one that we use in Switzerland is set up for looking at very small things. So typically the biggest specimen you could put in would be about 2 centimeters cubed, but normally we'd be looking at things no more than half a centimeter or so. But there are other similar facilities that can do X-ray tomography on much larger things, and in fact and then all the way through to sort of medical grade CTs, or computed tomography scanners that can scan entire skulls of dinosaurs, or even entire cars or engine parts. So there's a huge range, and it really is a case of choosing the right tool for the job.
Now, your answer on the Silurian earlier has caused some dissent among the chat, and there's a lot of people speaking up for the joys of the Cretaceous. Think of that what you will. A brilliant question that I think I haven't seen before and deserves an answer is from Helen Bennett, who wants to know, do the woody parts of plants count as skeletons?
Oh. So that's a brilliant question. Yeah. So there's kind of the broad definition of a skeleton, which would be any inorganic component that is laid down by an organism. So that is of a hard skeleton. So in that case, that they wouldn't qualify, as it were, because there's no mineral component like there is in say the shell of a snail or in a bone. But then there's a broader definition of skeletons as to be anything that supports the body using big molecules, macromolecules, outside of cells. So you can have this soft extracellular skeleton. In that case, they would, I suppose, be defined as a skeleton, but you'd have to ask a botanist, I'm afraid. But that's a really great question.
And we'll go over to a question from Roger Caseby, who asks, could changes in the modern ocean make it more difficult or easy for extant species, those ones that are with us today, to mineralize tissues? So could changes in the modern ocean make it more difficult or easy for extant species to mineralize tissues?
Yeah, so hi Roger, thanks for that question. Yeah, absolutely right. So the chemistry involved in building these skeletons is very finely tuned to the organisms and their environments, and we see in the fossil record that the prevalence of different kinds of skeletons made of different kinds of minerals waxes and wanes with different climatic conditions. And we know that changes in pH has a fundamental effect on organisms that build their skeletons. If the pH gets too low, becomes too acidic, then the ions that they need to build their skeletons, they can't get hold of, and they can't do their skeleton building. So absolutely, if the ocean continues to acidify, then there's good evidence that animals will have a harder time to build the skeletons that they need. Absolutely.
Now, at this point, I'm going to give you your second highly controversial question of the evening, or rather, very good question, but no doubt a highly controversial answer. This is from William Sharpley, who asks, what's the oldest animal?
Well, hi William. Yeah, so that's definitely a question that would probably have been better posed to Frankie, who would have given you a very different answer perhaps next week. Now I agree actually with my colleagues that the evidence for the oldest animals that we're sure we have is is almost certainly among these ediacaran creatures, things like Charnia. But the as I said before, the molecules suggest that animals may go further back in time than that. So it really depends on how you want to define the question. So if we're talking about the oldest animal that we can place into a modern recognizable group, then they probably fall inside the small shelly fossils. But if we're talking about the oldest evidence for animals, then there's trace fossils, and then of course the ediacaran fauna are animals that are much harder to place in the tree of life. But so it remains an open question.
And while we're on controversial fossils in the ediacaran, in the Precambrian, we've got a very specific one from Kate Moore, and Kate asks, and you might need to explain some of the terms in this question. Have the sponge zygotes found in the ediacaran of the Doshanto Formation been disputed? So just for everyone at home, the Doshanto Formation is a Precambrian ediacaran deposit in China. And do you want to start by telling us what a sponge zygote is, Duncan?
Yeah, so that's a brilliant question. Yeah, so one of the records that I haven't actually mentioned at all in this talk is occasionally we get soft tissue preservation in 3 dimensions preserved in when you get the soft tissues become mineralized themselves. So these kinds of fossils are most famous from the Cambrian, of little tiny arthropods, and they're almost always seem to be the very early developmental stages. So essentially eggs, embryos, and larvae. And the record of these kinds of amazing deposits continues down into the ediacaran. So there's this Doshanto Formation in China that you mentioned, where you get these 3-dimensional fossils of these little balls of cells that look like they're dividing embryos. And there's been a waxing and waning of opinion about what these things are. Some people suggesting that they're earliest animal embryos. Others suggested that they're completely artifactual, if they're not organisms at all, and others suggesting they're giant bacteria or other things, similar things like that. So I think the prevailing opinion at the moment is that you aren't necessarily able to place them within animal groups, so firmly within the sponges, but the jury's still out on that one.
And while we're around the Cambrian explosion, we'll have a question from Anthony Bates, who asks, is there a specific set of environmental conditions that may have prompted the Cambrian explosion?
So there's a whole host of different things that are taking place in this interval. So we have very good, relatively good records of the chemistry of the ocean and the atmosphere, and that's getting better and better all the time. And we know that during this time, most of the rocks or most rock sequences that preserve this period of time don't have any rocks actually deposited from the very earliest part of the Cambrian, because it was a period when sea levels were rapidly rising. So we have this so-called great unconformity that was has been mapped very extensively, especially in North America, and very few places where you get the actual deposition. And this raising of sea levels probably did 2 crucial things. One, increased the available space, so lots more shallow sea, which is where we think lots of these organisms were evolving, but also eroded the land and washed the minerals required for building skeletons into the ocean. And it was probably also a time of warming. So we have these periods of glaciation earlier in the Ediacaran, and it's very hard to figure out the climate this far back in the past, but it's thought that it was probably relatively warm as well. So it does seem to be that you have this combination of different environmental factors to and this big increase in the available space to build to be an animal, and also the available minerals to start to build skeletons. So that's probably what was one of the contributing factors.
Just while we're talking about the Cambrian explosion, I'll just remind everyone that in 2 weeks' time, Professor Paul Smith, director of the museum, will be talking on the topic of the Cambrian explosion and when it happened and what happened and perhaps some of the causes. And the link for that should be up on the chat now. So do register for that, and then it will be in your diary. We're going to zoom through some more brilliant questions. We've got one from Robin Saunders, who asks, does the toolkit hypothesis suggest that each small group, for example, on your diagram, each small group evolved the same skeleton type multiple times in parallel? Or is it possible that each group inherited their common skeletons from a shared ancestor?
Yeah, that's a really important distinction. So I think that it depends on the scale of the groups and how you're sort of defining them. So I'm pretty confident that within, for example, the mollusks, there are a number of different subdivisions of mollusks that have biomineralized skeletons, and I think within mollusks the skeleton evolved many different times, but probably within a group like the bivalves, there's probably a single origin from what we can tell, sometime around the origin of the group themselves. So it seems that at some point you get this innovation, which is then locked in, as it were, into this group. And now obviously there are examples where organisms have then gone on to lose those skeletons, but very often it's a very highly conserved feature. Once you've gone down the route of being something that needs to have a biomineralized skeleton, it tends to be retained. So I think there's a bit of both depending on the scale that you're looking at. I hope that makes sense.
So we're going to take two questions together here. I think two quick-fire questions.
Okay.
The first is from Lara, who says, can you recommend a good book on early life? And alongside that, I'm going to put Siddharth's question. Can you tell me the name of the first cellular organism on Earth?
Oh, goodness me. So the answer to the second question, I think, is no. I don't think I can. But that's probably because nobody knows, because it's almost certain the case that the very first of whatever group of animals that you're interested in almost certainly never got into the fossil record, because there's a relatively small chance of ever getting fossilized. So I'm going to have to dodge that particular question. In terms of books, I mean, if you really want to get into this topic, there's a fantastic book by Doug Irwin and Jim Valentine called, I think this is called, The Cambrian Explosion. It's probably one of the ones behind me on this bookshelf. But there's a whole host of other sort of popular science style books. I mean, going back to Steve Gould's book Wonderful Life, that sparked the interest in this topic in a whole generation of evolutionary biologists and other books. So yeah, that's where I would suggest you'd start.
Brilliant. I'd just say, in addition to what Duncan said in response to Siddharth, if you look at the very early cells preserved in the rocks of the Pilbara in Australia from about 3.4 billion years ago, only some of them have been given scientific names, and some of them are just documented, and they unfortunately go without a name, which is very unfortunate. We now have a question with perhaps a slight conflict of interest. It comes from a certain 5-year-old called, which has been put forward by Joe Murdock. So I think this is from one of your children. And I think this is a highly profound question that has both different geological and legal answers. Is a rock a fossil?
Oh, wow. Well, so no sweeties for you tomorrow for giving me such a difficult question. No, of course not. No, it's a brilliant question. Thank you. So we define a fossil as any evidence of a living organism that makes its way into the rock record. So whether that's its soft parts preserved in these wonderful ways, or shells or teeth or bones or burrows or trackways left behind. And in many cases, rocks can be composed of the hard parts of organisms. So chalk that was mentioned earlier in the Cretaceous is essentially composed of fossils of these single-celled organisms called coccolithophores. But there's also examples of rocks that are composed almost entirely of bivalve shells or crinoid ossicles, or so in some instances, yes, that the rocks can be composed of fossils. But in terms of most rocks that are built, they're created in processes that have nothing to do with directly to do with life, then they wouldn't be described as a fossil in the strictest sense. Thanks.
And we've got a wonderful conodont question from Adam, and there's lots of conodont fans in this evening. The moment they went up on the slides, we saw leaps into jubilation from
fans.
Megan and Brian and Chris and Will, all going very happy for the conodonts. But this question is from Adam. Are there mineralogical differences between conodont basal bodies and the crowns, or are they composed of the same form of calcium phosphate?
Okay, thanks, Adam. Yeah, so these two different tissues that I talked about, the blue and the red, they're different in the way of that the crystals were laid down. So in the crown, it's what's known as a hypercrystalline tissue, where it's essentially just crystals of fluorapatite, whereas the basal bodies are more of a composite between the mineral and an organic component. So they are often arranged in a fundamentally different way. A good sort of analog is the difference between dentine and enamel in vertebrate teeth, modern vertebrate teeth. But yeah, so there is a distinction.
So we're going to move into our final few questions now. Just a reminder, if you have any thoughts on the question that is on the slide now, has this lecture changed your opinion on the evolutionary origin of animals or added to your understanding of the topic? If so, how? Do please put your thoughts in the chat. We'll go to our final few questions. The first one is has just been, actually, I'm going to save that one until later. And I'm first going to go to Daniela, who asks, what's your opinion on there being an explosion of preservation during the Cambrian explosion rather than explosion of life itself? Okay.
So I think that there is definitely a component of that, in that there are these huge phosphate deposits, for example, where we get abundant small shelly fossils. And there is this change in the sedimentology, whereas we have this big erosive event. But there are many places in the Ediacaran, and indeed in much older rocks where you could easily have preserved fossils, like typical sort of fossils. And in fact, there are older than the Cambrian, there are deposits where we get phosphatic microfossils that are clearly not animals, that are derived from something else, from a single-celled creature. And we also get non-animal soft tissue preservation of things like algae prior to the Cambrian. So there is definitely we should see these animals if they were there. So there's part of the signal is a change in preservation, which fluctuates throughout the whole rest of the record. So whenever we're talking about changing in diversity or abundance of different fossil types, we must always think about the propensity for that organism to get preserved. As I say, it's changed. There were other periods in Earth history that are very low in fossils because of the conditions to preserve them weren't there. But it can't be said that we can put the lack of Cambrian type organisms in the Ediacaran down to preservation.
Now, in addition to a great deal of praise for your lovely shirt, Duncan, we have
thank you very much.
A wonderful question here from Sienna, who asks, would it be possible for a Cambrian explosion to happen in the future, given that we now know about genetics and new technology? Could there be a second Cambrian explosion?
So that's a really great question in a kind of from a sort of philosophical perspective in some ways, and there have been throughout subsequent Earth history, and I'm sure there will be again, major so-called adaptive radiation. So things like the origin of land plants, or then subsequently flowering plants, or winged insects, or flying dinosaurs, birds. But none of them are really on the same scale as the Cambrian explosion, really. The conditions of the Ediacaran have never really returned wholesale on to the Earth, really. So once you have animals doing their things, churning up the sediment, moving into the water column, it really has totally changed the way the whole Earth life system actually works. So I think almost certainly in the future there will be new forms of animals that evolve and adapt to new environments, but it won't be the kind of major fundamental change that we see in terms of animals in the Cambrian explosion.
Wonderful answer, looking to the future, and I'm going to give sticking with that theme of looking to the future. Okay. I'm going to give the final question to Jules Bristow, who asks, would you want to become a fossil?
Oh, that's something that I have actually thought about before. Where's the best place to end up to become a fossil? I mean, some people think that I already have become a fossil. In fact, I get that criticism, but I think the chances of getting fossilized are extremely small. So I heard somewhere, and then I'm sure it's more or less true, that you can put the entire fossil record of human remains in the back of a Land Rover, because they're so vanishingly rare that you get the actual bones preserved in terms of in deep time. So it's very unlikely. If I could make sure that it happened, then perhaps I could. I'm sure we could all imagine some entertaining positions or places we could end up to confuse future paleontologists, whatever they might be. But thinking from a sort of environmental perspective, I think it's also important that we remember the impact that we as humans are having on the planet as well, and in terms of leaving our legacy behind. We're all creating fossils all the time, in terms of the amount of concrete that we're putting in the earth, the amount of metal and the amount of plastics, radioactive elements. So in these so-called techno-fossils, so in that way, we've all already left fossils behind every time we're using any of those kind of things. So those sorts of fossils, I think it would be better if we didn't. But so maybe that's something to think about. But thanks a lot.
There you go, everyone. A profound moment to end on. Think about the fossils that you're leaving behind. Thank you again to Dr. Duncan Murdock for a wonderful talk.
Thanks, Jack.
And answers to a wide array of questions. Thank you, everyone, for joining us this evening. It may be this evening here, but I'm sure it's very different times from you all around the world. Thank you for joining us. And we will be back again in 2 weeks' time, when Professor Paul Smith will be talking on the Cambrian explosion and his view on that, based on the fieldwork that he's done in northern Greenland. And as I've said previously, there are going to be some very beautiful field photographs, as well as some incredibly exciting science there. So do join us for that in 2 weeks' time. This series of lectures will be continuing throughout the summer and the early autumn. We'll be making an announcement on speakers soon, but we've got some exciting people lined up, and we hope you can join us. But at that point, I will just simply say thank you once more for joining us, and we very much hope to see you in 2 weeks' time. Thank you very much.
Thanks very much. Bye, everyone.